So we're going to cover our next phylum very quickly, which is a phylum Placozoa. It has a single named species, Trichoplax adherens, but there are probably several cryptic species. Genetic studies suggest that there are at least several different genetic types, but externally they're hard to differentiate, so they haven't really actually been named. But they're basically small, large specimens get up to between 2 and 3 millimeters in captive and sort of cultured populations, but in the wild they're mostly actually smaller than that, typically around 1/2 millimeter or so in size. They're flat, basically 2 layers of cells, upper and lower layer. They're asymmetric, they have no obvious symmetry, and the edge of their body can change shape relatively fluidly. They're sort of vaguely, roughly roundish, disc-shaped, but they can, if you look at them, they can look kind of like amoeba-like in that they can change their shape a little bit, but they don't extend out to true pseudopods like an amoeba would. You don't get the kind of flow of cytoplasm that you would see in an amoeba, for example. They're found in marine environments. They seem to be crawling over substrates. The very first specimens were actually recovered from a commercial public aquarium from some of their tanks, so they kind of get caught that way. So they're basically usually a large individual will be several thousand cells arranged in 2 layers. There's an upper layer and a lower layer. These are relatively tall columnar cells, especially in the lower layer. In between, there is a mesenchyme, which is basically which is somewhat gelatinous. It contains fibers, protein fibers, collagen fibers, as well as amoeboid cells that can move through that mesenchyme. So it's not completely acellular. None of these areas truly are, but it is not. But the cells are mobile in there. There's not a fixed cellular structure in the intervening area. So there is a differentiation of cell types between a dorsal and ventral layer. The ventral cells tend to be larger, more column-like, columnar, and they tend to be secretory, and that is they are able to secrete compounds, molecules like digestive enzymes, for example. Both layers are covered in flagella, which are often more prominent on the lower level, and those flagella are basically used to glide over surfaces. So this is a small little flagellated disc that kind of glides around over rocks and shells and things like that. One key feature is that the ventral surface can be invaginated. That is, the cells can kind of push upwards. This is an exaggeration here. It's not quite that extreme. But the ventral cells can push upwards and create a digestive chamber, and this would be more efficient. So if you're crawling over something that you want to eat, you can just kind of secrete digestive enzymes, but they will kind of disperse out, and so it's relatively less efficient, especially in an aquatic environment. This is how fungi feed, for example, but they're in a terrestrial environment, so their enzymes can't really disperse very far. In an aquatic environment, they can. So by forming a digestive chamber, you can trap a small food particle in here, and your digestive enzymes will be concentrated in that spot and be more efficient mechanism of eating. And then the cells, through phagocytosis, would consume food particles. There are some receptors that among some of the cells that are similar to neurotransmitter receptors found in cnidarians. So these are cells that can, these neurotransmitters are molecules that basically transmit a signal from one electrically charged cell to another using a chemical pathway, using a chemical signal between those 2 cells. And the receptors are the cells are found in a receiving cell, basically saying that the other cell just received an electrical impulse and usually triggering an electrical impulse. Now, we don't have direct evidence for these kinds of, actual nervous system here, but they do have some of the receptors found in cnidarians. These receptors haven't been located in sponges, and so that suggests that these are one of the little bits of evidence that suggests they're a little bit more derived than the sponges. In terms of reproduction, it is actually very important. They've been observed to bud, so a small individual can kind of pinch off of the adult. But more importantly, they've also been identified as producing eggs and sperm cells, and so are capable of sexual reproduction. The reason that's important is because it actually tells us that these things are mature adults, if we can observe them reproducing, as opposed to perhaps being a simple larval form of something that might otherwise be much more complex. So these are, in fact, what the animals look like rather than just being a larval stage, because lots of larvae are kind of simple disc-shaped things as well. It wouldn't be surprising if this was a larvae, but since they're capable of sexual reproduction, that tells us they're an adult species. And that's about all we're going to say. In terms of the genetics, they have relatively small chromosomes, relatively small amounts of DNA. They're actually more similar to protists in that regard. The DNA sequence evidence generally suggests that they're located between the periphery, in terms of phylogenetics, they're located between the periphery and then Cnidaria and the Eumetazoa, Cnidarians and bilaterally symmetric animals here, basically there. So they're more derived than the sponges, but they're sister group to the Eumetazoa, to all of the other animals. Now there's one group that's not in this phylogeny, that is the ctenophores, and we'll talk about, because they could be all over the place, so we'll talk about those when we get to the ctenophores. And that's basically it for the Placozoans. I think that's my last slide here. Yep. Okay. Now, we are very, very slow today. All right. So our next phylum was actually the next one on that phylogeny over is a phylum Cnidaria. We already seen these in lab. Now we'll get to talk about some of their details that we didn't necessarily get to see in lab. Before we get to the Cnidaria, just in general, the Cnidarians are the first of the phyla that belong to the clade Eumetazoa. Basically, the Eumetazoa are all of the, so remember, Metazoa is basically is the same as Animalia, the animals. Eumetazoa, the sort of true animals or good animals, will basically be everything besides sponges and placozoans. So Eumetazoa are characterized by having true tissues. Those true tissues include specialized cell connections that connect those cells together in various ways. Excuse me. For basement membranes, it means having an for epithelial tissues, it means having a basement membrane upon which they sit, and among other things. And cells generally fixed in the type and where they are. So you can't really have true tissue and specialized cell connections if your cells can get up and move around, if they can change type and so on, like they can in the sponge. In all of the Eumetazoa, the tissues are in the embryo during early development. Those tissues are organized into first 2 layers, and then in many Eumetazoa, 3 layers. That is, they're either diploplastic or triploplastic. Obviously, for Cnidarians, they're going to just be diploplastic. And all of the Eumetazoa, unless it's been secondarily lost, have specialized muscle and nervous cells capable of electrical stimulation. So we have some contractile cells and sponges, and we have some probably have some contractile cells and placozoans. But in the Eumetazoa, we have true muscle cells that are stimulated via electrical impulses, and we have nerve cells that can transmit electrical impulses. So those are specialized tissues. Those are 2 of the specialized tissues found in the Eumetazoa. And then the connection between nerve and muscle cells, or between nerve cells and nerve cells, are called synapses. And they can be sometimes a direct electrical connection. More commonly, they're actually a chemical connection. That is, the leading cell basically produces a chemical signal that diffuses across a very small gap, the synapse, and stimulates the downstream cell. All of the eumetazoa, again, unless occasionally secondarily lost, have some sort of symmetry, either radial or bilateral. And again, unless it's been lost, they have some sort of a digestive cavity, more so than a permanent digestive cavity, as opposed to that little hollow space that you get in the placozoans. All right, so the first of the phyla of the eumetazoa are the Cnidaria. These are jellyfishes, hydroids, anemones, corals, and things like that. You may occasionally see the group referred to as a Coelenterata. That's an old term that basically combines them with the superficially similar ctenophores. But the ctenophores and Cnidarians are probably not that closely related, although there's a lot of debate about their relationships. And so we don't really use the Coelenterata much anymore, but you might still encounter it, in which case it's Cnidarians plus ctenophores. They're currently recognized over 13,000 species of Cnidarians, so a reasonably diverse group. They are diploblastic. They have an epidermis, an outer layer derived from the embryonic ectoderm, and a gastrodermis, remember gastrostomach, which is a layer that lines the gastrovascular cavity derived from the embryonic endoderm. Between those two layers is a mesoglia, which is, again, mostly acellular. You can have some motile cells come in there or extensions of cells, but it mostly tends to be acellular in the Cnidarians. Cnidarians exhibit radial symmetry with one exception, or one genus I think is an exception. They have radial symmetry. They have different types of radial symmetry, however, so you can have sort of quadriradial symmetry. There are 4 planes of symmetry here, 1, 2, 3, and 4 ways to divide that in symmetric halves here. Some have 6 and 8-fold symmetry, so they're but as well, and some have just some sort of generic radial symmetry. And they seem to, and they prefer that. So recent studies with jellyfishes, for example, have found that if you chop off a hump, they can recover from an injury. But if you chop off a hunk of a jellyfish, it won't be radially symmetric anymore. It won't be symmetric anymore. And so they actually rearrange their body parts and muscles and things to kind of reestablish radial symmetry. So these are animals that try to be radially symmetric. All right, as we've already covered in lab, they have a gastrovascular cavity that is a digestive tract with a single opening that functions as both a mouth and an anus. So they take food in, and then waste products are expelled out the same. So basically, they can vomit, but they can't poop. Think of it that way. And that mouth is typically surrounded by tentacles, long skinny extensions of the body. The body exhibits 2 basic forms, a polyp form and a medusa form. Polyps are usually somewhat column-shaped. They're typically, again, there are exceptions to a lot of these rules, but they're typically sessile and attached at the base of the column, and the mouth faces upwards, surrounded by its tentacles here. In the mesoglia, which is shown in red in these diagrams here, so the pink on the outside is the epidermis. The tan here is the gastrodermis lining the digestive cavity, and then the red in here is the space between it is the mesoglia. In contrast to the polyp, the medusa is generally free-swimming. Again, there are some exceptions. We'll cover some of those. But generally free-swimming with the mouth facing down. The mouth may be extended downwards from the tentacles here. And the mesoglia is often typically much better developed. It's thicker and more jelly-like, acting as basically a hydrostatic skeleton in the medusa forms here. And I should point out that the mouth is, so in animals that are radially symmetric, the side with the mouth is called the oral surface. The opposite end would be the aboral surface or aboral end down here. So this is aboral here, and then this would be the oral side here where the mouth is. Okay. So one of the key synapomorphies that unite all of the cnidaria and is the presence of cnidae. Cnidae are actually organelles, and so the cells that support cnidae are called cnidocytes, site meaning cell. And so the cnidae are specialized organelles. You might have heard of the term nematocysts. Nematocysts are one kind of cnidae. They're actually lots of about 30 different kinds. Nematocysts are one of the more typical types of cnidae that are stinging. So the cnidae develop from within specialized vacuoles. There's some suggestion that they might actually be of endosymbiotic protist origin. That is, they derived from a protist that might have been derived from a protist that was then captured and modified into an organelle. There's debate about this, and we don't really know what the protist would have been, but they're certainly very unique and distinctive structures, and they're found only in the cnidaria, or in animals that have eaten cnidaria and somehow captured their cnidae. So the cnidae, they're generally pretty small, usually less than 100 microns in size. So remember, that's about 1/10 of a millimeter or smaller, so usually essentially microscopic in size. And they can be pretty numerous. They're usually abundant around the mouth and around on the tentacles, but they can cover the entire animal. Basically, the cnidocytes would be just scattered amongst the other epithelial cells, epithelial cells, other cells of the epidermis, basically, and they reach the highest concentrations around the mouth and tentacles. All right. So basically, what are these cnidae like? Well, they're basically basically here's about. So this one is labeled nematocyst. So this is specifically a nematocyst. So the cell here, in the sort of stippled box here, is the cnidocyte. That's the cell that contains the cnidae or nematocyst. There's the nucleus of that cell, and there's a large vacuole. And inside that vacuole, you have another layer of membrane here that is basically turned inside out. And then there's a lid at the top, at the edge of the cell here, called an operculum. That term, you've also already seen that term, right? That's just a commonly used term for any kind of a lid, basically, or covering. And so what happens is that as pressure builds up in this vacuole, this cnidae actually turns inside out, and the insides here basically get pushed outward. So again, it's like you have that, think of a glove with a finger pushed inwards into the glove, and then if you squeeze the glove, the finger pops out, and that's basically what's doing it. So this would be the inside of the glove here. The finger is basically pushed in this way and all kind of folded in on itself, and then it can shoot out, and they can be long and skinny or fairly short. They have a wide variety of structures. You'll also notice this little thing sticking out here is called a cnidocyl. We'll get to that in a second. All right, so cnidae are actually variable. Nematocysts are just one of the stinging type, but there are lots of different types of cnidae. There have been 30 different forms identified. So some of these are long and skinny and just kind of wrap around the prey. Others are kind of have little sticky hooks and barbs that adhere to prey. Some can be actually sticky. Some have little hooks that will stick to prey or even objects. Some are kind of have a sharp, big hook that can pierce the prey, and some are capable of injecting venom. So you have piercing cnidae, you have piercing and injecting cnidae, you have sticky ones that adhere. So for those going to San Diego, for example, we'll see clusters of aggregating anemones, for example, which are anemones are a type of cnidarian, and you may actually end up walking on them without even realizing it, and wondering why the rock is kind of squishy. And part of that is because their walls, they will stick all kinds of little sand and shell fragments and so on to their walls, to their sides, and then the tentacles, if they're out of water, are just drawn in. So they don't look, it just look like a massive kind of gooey shell fragments. And they're using these not specialized nidae with adhesive things to stick shell bits and sand and debris to them, to their column as kind of camouflage and a little bit of protection. So they can do a lot of things besides just stinging prey. They also use them to fight each other. They actually have specialized, many anemones, for example, have specialized tentacles that they use for fighting amongst each other compared to those that they use for feeding. All right, so basically these nidae are kind of cocked in their cells. When they're discharged, these tubes shoot out at about 2 meters per second. This is a very, very fast. This is one of the faster cellular. Remember, this is happening. These are structures within an individual cell. So this is one of the fastest sort of cellular processes known. The exact mechanism of how they're discharged isn't actually known. It's very likely that it involves hydrostatic pressure. So basically, the pressure in the cell or in the vacuole around it, in the nematocyst around the inside part here, builds up, and that's what pressure, once it's released, basically ejects it outwards. What we don't really know, however, is whether there's a sudden increase in hydrostatic pressure by, for example, opening lots of channels and allowing water to flow into the cell, or if they're generally continuously under pressure. And then what allows them to shoot out is they release, basically opening that operculum, and then they just release that pressure. So the details of exactly how these shoot out aren't actually well understood yet. One key feature of these nidae here, and again, this is a whole nidocyte. Here's the nucleus. Not sure what this curly bit here is. And then you can I think, see a little bit better. There's a cover, an operculum, and you can basically see this kind of, this is the big vacuole here, and then you have this tube, which is basically coiled up inside, but inside out, so that when you release the pressure, this whole tube gets inverted inside out and shoots outward. One of the key features is, I pointed out earlier, but here it shows a little bit better, is a nidocyl. A nidocyl, cyl is short for cilium, and it's basically a modified cilium that's stiff and responds to pressure or touch. So when something bumps up against the edge of a cnidarian, the caustic tentacle, it will bend this little nidocyl, and that nidocyl will trigger then some sort of an internal response that causes it to shoot up. So this is one of the main mechanisms that activates it. They are activated in response to touch. So a tentacle brushes up against a little prey item, brushes up against the tentacle. These guys activate, and the little nidae shoot out and stab the prey item. Same if a potential threat brushes up against them as well. They can also be triggered by chemical cues, and there's probably some nervous system control, although again, that's not particularly well understood at this point. All right. So as I pointed out, so some of these, some nidae can contain venom, particularly those, the nematocysts in particular are the venomous type. Those venoms typically tend to be neurotoxins, which means that they disrupt muscle and nerve function. That's particularly helpful because it A, can be very painful, which may inhibit a predator from going after you. But it can also subdue prey, which would otherwise be moving around and crashing and fighting back. But if you can disable its muscles and nervous system, that prey is much easier to subdue and is no longer dangerous to take into your soft, squishy body. And some of those neurotoxins are strong enough to affect humans. Particularly some of the various jellyfishes have relatively strong toxins, and nematocysts that are strong enough and thick enough to also penetrate our skin. So, to get stung by a cnidarian, you have to have a strong enough toxin, and you have to have nematocysts that can fully penetrate skin. So some jellies do that. The so-called fire coral, well, not so-called, the fire corals, which get their name because of the burning sensation of their venom. Portuguese man-o'-wars, which can be potentially deadly to humans if you get tangled up in their tentacles. And a group of jellies, class we didn't see in lab, are the box jellies, also called sea wasps, and which are actually really cool, and we'll talk a little more about them later. In the class Cubozoa, and they can also be extremely potentially deadly as well. A single cubozoan may not necessarily be deadly, but they can often be in swarms, so you get stung by hundreds of them. And their neurotoxin acts pretty quickly. And if you're in their habitat, that means you're probably out there swimming or something like that. So being immobilized is usually not a good thing for us air-breathing humans. So there is some evidence that the nematocysts can be suppressed by acids or bases. But which one, what effect it has depends on the species. So vinegar works, at least for some kinds of nematocysts. Baking soda, which is more of a base, works for some others. Urine is supposed to work because it's somewhat acidic. I forget, but it doesn't. It can actually make things worse because basically what happens is if you get you brush up against a tentacle of a jellyfish, some of those nematocysts, those cells and those tentacles might be stuck against your skin, but not all of the nematocysts have been discharged. So what happens is they may get triggered over time and make the pain worse. So an acid or a base basically potentially suppresses that discharge, and therefore doesn't make things any worse. It doesn't really make the pain go away, but it reduces future discharges. The problem with urine is that in most cases, unless you're really dehydrated, your urine is actually much more dilute than seawater in most cases. And very dilute water and fresh water can actually trigger nematocysts to fire, so it can actually make things worse. So, if you get stung by a jellyfish, there's probably not any real benefit to getting peed on, unless that's something you're into. Okay. So, as we talked about the body form, so medusae are almost always solitary. There are perhaps a few exceptions, but they're largely solitary. They're almost always free swimming. They're generally much less diverse in their structure than the polyps are, so they all tend to look pretty similar. They generally are bell-shaped. They can be a little bit broader or taller, but they still have a general bell shape with the mouth facing down, the oral surface facing down. The mouth often has a tubular extension, you can kind of see right in here, called the manubrium. That's fairly typical of most of the medusae forms as well. And most forms, regardless of which class they belong to, tend to have quadriradial symmetry. So they often have, structures arranged in groups in sets of four. So, a jellyfish will have 4 gonads and 4 digestive glands and 4 oral arms and things like that. Okay. Polyps, on the other hand, tend to be much more diverse in their size. They're usually pretty small, but especially for the anemones, and even a few corals can get pretty decent. There are some anemones which can extend themselves up to about 1 meter tall, so it's about something like that. But they can also shrink down. These animals that are mostly filled with water, and the anemones extend outwards often by just filling their gastrovascular cavity and inflating that way. So some anemones can get pretty big, but most polyps typically tend to be quite small. They typically have either quadriradial or biradial symmetry. Some sort of radial symmetry. Quadriradial and biradial is present in some. And one somewhat worm-like genus of anemone is technically bilaterally symmetric, although the picture here doesn't really show exactly where its symmetry lies. So can't go by the picture too much, but that's what it supposedly looks like. Otherwise they're except for that one exception, they're basically radially symmetric. And polyps are usually sessile, that is, they're usually attached to the substrate. I mean, they can move around, they're not necessarily fixed, but they're not swimming or moving. There are some exceptions, like some of the siphonophores that you guys saw in lab, like the Portuguese man of war, are technically polyps that are mobile. But for the most part, polyps tend to be sessile, like this anemone here. Part of the reason for the great variation in structure among polyps is that polyps can be solitary, like these guys over here, but they can also be colonial, depending on the species. They can form colonies. So here, for example, is a gorgonian, a kind of soft coral, and you can see each individual polyp here, and those polyps are interconnected through a larger sort of skeletal structure that they secrete and kind of contribute to. They may actually join up their gastrovascular, they typically join their gastrovascular cavities so that a polyp that eats something here can provide nutrition to its nearby neighbors. So here you can see all of the polyps look pretty much the same, but in some cnidarians, the polyps can have different types of polyps. And I should point out that polyps are produced through asexual budding typically. And so all of these polyps are basically genetically identical. That is, they're all clones of whatever the polyp was first. So these are, in a way, you can think of these sort of colonial animals as not so much a colony of separate individuals, but as a single animal that's built on a more modular design. That is, rather than having an animal that's built up of lots of complex parts that extend through the whole animal, they're made up of lots of smaller, simpler parts that just repeat again and again. Okay, so I think I may have just said most of that. So yeah, colonies formed by asexual budding. Each member of a colony, this applies not just to cnidarians, but other colonial animals as well, but each member is called a zooid. So each of these little guys, this would be one zooid, this would be another zooid here. And like I said, I already mentioned, polyps are connected by and share a common gastrovascular cavity in most cases. They can be encrusting, that is, they can grow kind of on the surface of rocks and shells and things like that. They can be kind of boulder-like, creating, especially if they have a calcite exoskeleton. They can be branching, they can form branching structures like this, so they have varied structures. And they're basically kind of a modular organism. You can just keep adding extra little units here. And so the colonies themselves, even though the individual polyps, the zooids, are very small, the colonies themselves can become very large. All right, where are we on time? All right, so colonies of cnidarians can be monomorphic. That's the example I just showed you. Is a monomorphic colony. There's a single type of polyp or zooid, and they're all basically the same. So that gorgonian I just showed you was an example of a monomorphic polyp, all of them the same. Most stony corals also have monomorphic colonies as well. But some species have polymorphic colonies, which means that the polyps are different. They have different types of polyps. Remember, they're all going to be genetically identical. They're just turning on or off certain genes within each to become specialized. So you take that modular design, and now you have several different modules you can play with. Rather than just having one module repeated again and again, you can add a bunch of different modules, and they can have 3 different types of polyps, depending on the species. Gastrozoids are basically feeding polyps. They always have tentacles and a mouth, and are the primary feeding structures of gastric stomach. Dactylozoids are defensive polyps. They tend to have very powerful venom, and that is often very painful, so they can help to defend the colony, and sometimes may also help in capturing and immobilizing food as well. And then gonozoids are gono for gonad, are reproductive polyps that produce gonophores, basically, which are the next generation, and they may be for animals that have a alternation of generation life cycle, those gonophores may be released as medusae. Gonozoids typically don't have a mouth and are not feeding, so they're sort of specialized for reproduction. And dactylozoids may not have a mouth either. Okay. So here's an example. This is a colony of hydrozoans. This is a hydrozoan that grows on like the surface of seashells, I think. That's a shell encrusted one. So it's a very small. It would be like a little fuzzy crust. If you looked at it close up, you'd see maybe a little fuzzy crust on the seashell. And then you zoom up real close, you get a whole bunch of different polyp types here. So you have gastrozoids, feeding ones. Notice that they're surrounded by tentacles here. There's its mouth. But you also have gonozoids, which are basically these sort of sac-like, more sac-like structure, loaded up with the next generation. There's another gastrozoid, and then you have dactylozoids here, which you can see don't have tentacles, but they have these little pulses at the little bulbs at the end. Those are short little tentacles loaded up with nidae, with nematocysts that are highly venomous. So this is kind of one of the more protective zooids here. There's another dactylozoid at the end. So no tentacles or feeding structure, just a little mass of tentacles. Oops. There we go. All right. So one group of these polymorphic cnidarians are the siphonophores. We had a preserved Portuguese man of war in lab as one example. That's the one that's probably more familiar to people. There are various, so siphonophores are polyps, are colonial polyps, except unlike most polyps that attach to the substrate, they're actually free swimming. And so you have pelagic polyps. The polyps are generally polymorphic, and they can drift around. There may be a swimming bell, one or more swimming bells. Those are all. There's debate about whether, depending on the species, in some cases they appear to be a medusa form, the medusa generation that is attached to the polyps generations. In some cases, it may be, like in Portuguese man of wars, it's probably just a modified polyp that can basically inflate with water or with gases that basically help it to drift and up and maintain a positive buoyancy. So they may have one or more swimming bells or a float, which is generally a polyp form. Then they have gastrozoids and dactylozoids and gonozoids. The gastrozoids and the dactylozoids typically each bear a single tentacle. So even the feeding zooid has a single feeding tentacle. The dactylozoids will have a single, often very, very long tentacle loaded with nematocysts. And then the gonozoids are the reproductive ones that produce gonophores. So here, for example, you have a swimming bell, you have a float, that's probably a polyp, and then hanging from each of these, you have multiple polyps. You have a gastrozoid for feeding. You have, this one doesn't show a dactylozoid, the species might not have one, and then you have these sort of bulbous gonozoids, which contain the gonophores within them, the reproductive structures. And these reproductive structures, rather than being released, basically just become part of an ever-growing colony here that drifts around in the ocean. So they don't have a separate medusa stage, they all stay as part of a colony. And so this shows the different siphonophore polyps in, I think these are intended to be Portuguese man of war. Physalia, these are from Portuguese man of war. And so these show the three basic polyp types. So you have the gonozoid here, which has the individual gonophores that become the next generation. You have male and female ones here, so you can have sexual reproduction. And then there's a big blob here full of nematocysts for protection. Then you have the dactylozoid here. This is the main polyp and a single tentacle that comes off. And you can see how all of these little swellings here are literally covered with nematocysts. So you have a long seen tentacle, and that's basically all the dactylozoid does. And then you have the gastrozoid, which also has nematocysts, but it has a more complex tentacle with little branches here that it uses for capturing food. Again, they also have nematocysts for stinging their prey and then bringing it up to their mouth. But the key point here is that for siphonophores, as well as some other species of cnidarians, you can have polymorphic polyps. That is, you can have multiple polyp types within a single colony that are each specialized for different functions. So, all right, and so there's no sound here. The original video had sound, but it's actually better without the sound and some awful music. But this is a new deep sea siphonophore here, so it's actually a pretty complex thing. So there's floating bells up in this part. You can see a lot of the tentacles hanging down here. I would guess that those might be the gastrozoids here. I'm just guessing. All right, so moving on, another aspect of cnidarians is their structure. So cnidarians have mesoglea, so a gelatinous area between the two layers. This acts as a hydrostatic skeleton, primarily in the medusae, which have a much thicker, both a more gelatinous or denser, so a more jello-like mesoglea, but also much thicker as well. And so the muscles can act against that mesoglea to produce movement of the animal. So in medusae in particular, they rely heavily on the mesoglea for their hydrostatic skeleton. The gastrovascular cavity, however, is also filled with water as well. And so if you shut your mouth, well, if you're a cnidarian and you shut your mouth, you can basically act as a hydrostatic skeleton as well. Muscles acting on the cnidarian will compress. You have circular muscles that compress it, you extend outwards. If you have your elongated muscles shortened, then you compress, you get shorter and move to the side. And so if you have your mouth shut, basically all of the fluid inside you stays inside, so you're just rearranging that as you do with any hydrostatic skeleton. And a lot of polyps use their gastrovascular cavity essentially as a hydrostatic skeleton. In addition, some cnidarians have an internal skeleton of stiff rods, mostly made up of a protein and polysaccharide mix. This is used in some colonial forms, typically not in the polyps themselves, but in the supporting rods that supports all of those many polyps. So things like gorgonians, sea fans, and sea whips, and so on, like that branching guy I showed you a little while ago. That's a species where in the branch itself, there would be these sort of polysaccharide protein rods that make them relatively stiff. That would be an endoskeleton. Some cnidarians develop calcium carbonate internal sclerites. That is, they secrete these internal in their tissue, again, typically in the mesoglea. So some octocorals, which is one subclade, such as some gorgonians, may use these individual sclerites, kind of similar to the way a sponge uses spicules, and kind of the same principle here. You can also have an external skeleton, also of calcium carbonate. This is secreted by epithelial cells on the lower part of the polyp, And so it secretes calcium carbonate around the base of the polyp to kind of provide an anchoring point and protection for the base of the polyp. And since the bodies are relatively soft and thin, if you expel most of your water from your gastrovascular cavity, these polyps can shrink right down onto their exoskeleton. So this is obviously the most famous examples of these are the stony corals, the hard corals, but some of the hydrocorals that also produce a similar skeleton do the same thing. So here you're looking at just a single coral skeleton. There may or may not be a pedicel, and then there are a series of septa or ridges here that stick upwards, and the actual soft body would sit on top of this, and in between all of these septa here. And here's a anemone or coral in cross section. And so what you're looking at again, here's the basic column of the polyp. The mouth would be right here, the pharynx, and then the gastrovascular cavity is basically subdivided. There's a bunch of walls or mesentery that subdivide the gastrovascular cavity, and these are associated with these septa. So they sit in. So here's a calcium septa, and they basically sit around those, and the body extends down in between all of those cracks. And if the coral is withdrawn, for example, in response to a threat, it will pretty much be, it can drop down almost completely down into among the septa, among those ridges there, and protect itself. All right. Where are we in time? We're out of time, so I'll stop here. I'll talk a little bit about some of the threats to corals next time, and take a look at a little bit of Cnidarian diversity, and maybe start with, I forget how much we have left here, Tino 4 or something like that. Maybe we'll get to those.